This is a production of Cornell University.
The topic of my talk today is USDA apple, grape, and Tertiary Germplasm collection. I'm only going to give you kind of overview of what we do at Geneva. So if you have any specific question about any of my talk, or you have any question about the collection, please contact me later. First, very briefly about some of the history for the National Plant Germplasm System. President Lincoln established the U.S. Bureau of Agriculture, current day USDA, and also signed 2 months later signed the Morrell-Land-Grant College Act in 1862. And then in 1898, the section of Seed and Plant Introduction was established, and the first PI, which was cabbage from Russia, was introduced by Niels Hansen also at the same year in 1898. Also in 1898, the Federal Plant Introduction Garden, which is the first one, which is a current day subtropical horticultural station, was established in Miami, Florida in July the same year. And later in 1904, another plant introduction station was established at Chico, California, in Northern California. Later in 1919, two other one in Savannah, Georgia, and one in Glen, Maryland, was established. In 1946, the Research and Marketing Act allowed to establishment of 4 additional regional plant introduction stations, that including the Northeastern Project, we call it NE9, at Geneva, New York in July 1948, which has become current day the vegetable crop repository in Geneva. Here just showing you a diagram. This diagram is a little bit old, but basically give you some idea of the center of orange or center of fruit for a lot of fruit we consume current day. Except some of the small fruit is native to North America. Otherwise, majority of the fruit that we consume actually come from other part of the world. For example, apple is actually should be more central origin in this area. It's from Central Asia, and that's why we need to conduct exchanges, also exploration, trying to collect a lot of germplasm from other part of the world. Here are some of the pioneers that involved with the plant introduction. The first 2 persons on the top is David Fairchild and Walter Swingle. They are the 2 gentlemen in their 20s actually established the section of plant introduction in 1898. Niels Hansen is the first plant explorer. He introduced the first PI back into the US. And famous Frank Meyer spent a lot of years in China doing a lot of exploration. Wilson Popney, he was actually avocado plant explorer. Spent a lot of time in the highland of Mexico and Guatemala, and later in Central America and the Caribbean. Interesting part is he also a spy for the National Security Agent during the World War II. Later in the 60s, Professor Olmo, viticulture department at UC Davis, collect a lot of variety from the Mediterranean region. Of course, Dr. Charlie Ricks, Mr. Tomato, spent a lot of time in the Andes collecting all the wild tomato germplasm. A lot of those material actually also have a copy at Geneva current day. Here's just a diagram showing some of the location for a lot of germplasm stations throughout the United States. You can see we basically have a lot of different locations. Unfortunately, we used to have a location in Palmer, Alaska, and that station, due to the budget issue, was closed down 3 years ago. Some history about how the Geneva Colonial Repository came about. Basically, in 1974, there was an American Society Horticultural Science workshop on the topic of conserving fruit and nut crop. Was held in Gulf, Ontario, Canada in 1974. And later, Brooks and Barton, based on that meeting, they described a plan for a colonial repository that they argue that really have a different needs than the common seed repository. Therefore, from that discussion and planning in 1983, Bill Cermak, who is a supervisor for farm operation, was hired and came on board. And a year later, in 1984, Phil Forsline, who was the first curator, also came on board. For the apple, the Malleus, and also coral hardy grape, the Vitus, propagation actually started in 1983, and the first planting was in 1986. The Tertiary Collection was originally actually housed at the Davis Repository in California, but it was moved to Geneva in 1995 for better growing environment. In the USDA, our research unit actually divided into different units. We have 2 units at Geneva. First one is the Plant Genetic Resource Unit. Under that, we have 3 different project plans. The first one is the seed crop, or the vegetable crop. Second one is the clonal crop, apple, coral hardy grape, and tart cherry. The third is actually apple rootstock breeding program, leading by Gennaro Fazio. Then the other research unit is the Grape Genetic Research Unit, the GGRU. They're working on grape genetics and also genomic of grape. Here just showing some of the picture of the clonal collection throughout the year. This is a view from 1985, where basically it's about 2 miles from the Geneva campus. This is part of the McCarthy Farm. We occupy McCarthy South, and this is about 50 acres. You can see that picture taken in 1985. You start some nursery operation over there. 22 years later, this you can see at the same location, but just from opposite direction, the picture was taken. We basically have a lot of grape and tart cherry all the way in the back, and also a lot of apple. The slide below just showing a lot of different picture, diversity of material for the Malleus we have in our collection, and also some of the Vitus and tart cherry. They just have a gorgeous color during the fall season. The mission of the clonal repository basically throughout the entire system, basically a 4 basic mission. That's a similar mission for all the repository. Firstly is trying to acquire, second is trying to preserve, and the third is trying to characterize, and the fourth is trying to distribute all the germplasm we have. But each repository is a little bit different. For our acquisition, we're trying to get materials through exchanges and also exploration, basically on the worldwide basis. For our preservation purpose, we have a field collection of trees, and also we have a cold storage of seeds, most of wild germplasm seeds. Also, we use acquired preservation of dormant budwood at the USDA National Center for Genetic Resources Preservation Unit, Fort Collins, Colorado. From here on, we'll call them the Fort Collins. And also for characterization, we use a morphological method or genetic method, trying to study all the accession we have, and all the information from our study basically are stored at the Germplasm Resources Information Network database. We call that GRIN. A new version of the GRIN should be come on board this year sometime. It's called GRIN Global, supposed to have a better function, but it's quite a learning curve trying to change into the new system. For our distribution purpose, that's the major difference between the clonal repository versus the seed repository. Basically, our main distribution material are the dormant budwood, but we'll also distribute summer budwood. But for research purpose, we also distribute pollen, fruit, flower, fruit, seed, leaf, and also a lot of DNA sample in recent years, basically free to all the stakeholders and researchers, both in the United States and also worldwide. Also, another very important function of the distribution is trying to increase the public awareness of the value of the genetic resources we preserve. From the early on, basically as the establishment of the collection, because the location is at Geneva, so for the Malleus collection, basically we just knock on the door of Dr. Roger Way. That was the apple sign breeder at that time. We got all the donations from all the Geneva apple breeding program, basically been going on for over 100 years at that time. Get a lot of material from Dr. Roger Wei at the same time. But we also get a lot of material from other breeding programs in the United States, such as the PRA program. That stands for the Apple Breeding Program among the 3 campuses, Purdue University, Rutgers, and also University of Illinois. We also get a lot of material from other campuses and also from Agriculture Canada. We also get material from England National Collection at the Long Ashton Research Station, and also material from several different other countries. For the Vitis collection, basically we started by donation also from the Cornell grape breeding program. At that time was Dr. Bushreich, who is still in charge of the program. We also get some additional material from UC Davis, University of Minnesota, South Dakota, and several other places. Today I'm only have time to talk more about the Malleus wild germplasm material. Here is a diagram, a kind of map showing worldwide where the Malleus species distribute. As you can see, there are 4 different species in North America. There's 2 species in Europe, the Silvestris, Malleus silvestris, also Malleus florentina. There are 2 wild Malleus material in Central Asia, the Malleus orientalis and also Silvestrii. And as you can see in Asia, basically most in China, also some in Japan and other places, they just have more than probably close to 40 to 50 different species and hybrid species over there. Basically, in general, that's a consensus that Malleus actually originate from central and southwestern China. And then throughout history, especially through the Silk Road, material actually able to reach Central Asia, eventually went to Europe. And how the material came to North America actually was believed in the ancient time during the Ice Age. Actually, there's an ancient land bridge between the North America and the Asia. So the material actually came the other direction came to North America. And common modern apple we consume is the Malleus domesticata. It's a diploid species with a set of chromosome 34. It is strongly believed that the origin of modern apple actually came from Malleus silvestrii. There's also some parentage involved with maybe orientalis, maybe some silvestris. The question is still being debated. The next phase of the Malleus collection at Geneva I consider was expansion phase of the collection, especially through a lot of exploration activity from the late 80s into the 90s. The first exploration was Professor Whedon of Cornell University. He went to the west coast of North America to collect a lot of Malleus fusca in 1987. Later, the graduate student, Elizabeth Dickinson, for her thesis work, she collected a lot of material in eastern US, also in the Midwest. 3 different species, Angustifolia, Coronaria, and Ioensis. From 1989 on, Professor Herb Altwinkel, once again together with Elizabeth Dickinson, and also with curator Phil Forsline, they went to Central Asia, 4 different countries, to collect Malleus silvestrii 4 different times. Then in 1997, Professor Altwinkel, together with Phil Forsline, they went to collect 7 different Malleus species in China, in the southwest China, the Sichuan province. Later, in 2001, Gabel from Germany went to the same place to collect similar material. There were extensive exchange material between the 2 explorations. In 1998, Phil Forsline, together with Professor Amy Arizoni from Michigan State University, they went to Russia to collect a lot of material, including Malleus orientalis, also some other materials such as Tertiary and some other cherry material. In 1999, Professor Altwinkel, together with Phil Forsline, they went to Turkey to collect Malleus orientalis. Later, in 2001 and later, Joseph Postman from Covalis repository went to Armenia and Georgia, collect a lot of different species, including Malleus orientalis again. The Malleus silvestrii exploration in Central Asia has a very long history. A lot of Russian scientists, they studied the wild Malleus germplasm material in that region as early as I can find out was 1796 all the way into the 1900s. The famous plant geneticist Nicholas Ivanhoe Vavilov of Russia, he first explored the wild apple forest in Central Asia basically in 1929. And in 1929, Westover, who was a plant explorer, was sent by USDA to Alma-Ata, Kazakhstan. Translation means father of the apple. Also in 1929, met up with Vavilov and explored the material over there. And in the 1990s, Professor Danglov of Kazakhstan Academy of Science with his colleague, they explored all the wild Malleus germplasm material in Kazakhstan from the 1950 all the way into early 2000. There was an extensive review was written in 2003. Here's a picture from 1929. This is Westover, who came from USDA. This is Vavilov, and they met up in Alma-Ata, basically in Kazakhstan, looking around the Malleus silvestrii material in that region. And then basically 60 years later, another group of USDA scientists from Geneva went there to collect the material. Also, something interesting I dig up a little bit, Vavilov actually came to Ithaca in 1932 for the 6th International Congress of Genetics. He was the vice president for the society at that time. There's basically who is who in genetics. Both animal and plant genetics were all in that meeting, including Barbara McClintock. Also, in that trip, he also went to Geneva. He actually presented 2 paintings of wild apple from Kazakhstan and Kyrgyzstan to Professor Willington of Experimental Station, also in 1932. These 2 paintings actually still being preserved at the Lee Library in Geneva. I just wonder, anybody recognize the building on campus? Maybe it's still there, maybe not. So quickly, I want to make a pitch. Why do we need to collect and also preserve wild germplasm material from the wild? Because a lot of wild germplasm and also native habitat are lost due to agricultural development, also changes in agriculture practices. Also a lot of urban development issue, a lot of animal grazing, environmental degradation is a serious issue, such as the soil erosion, reduction in water supply and quality. Of course, the climate changes, such as greenhouse effect and desertification. Also, a lot of wild germplasm material also is being threatened due to new pests and diseases. Also, a lot of time, also invasive species into the native habitat. Also, a lot of time, plant biodiversity also being reduced because agricultural intensification, also agricultural commoditization. For example, for the worldwide avocado trade, basically 95% of all the trade just rely on single variety, the Hass avocado. Similar for when you go to most of the supermarket outside in New York, you probably only find about 11 apple variety. You will commonly see that in the supermarket. Chen from the Chen from the Chinese Academy of Science, they did a survey in 2009. They basically compare the native range of Malleus silvestrii in Xinjiang, China. Compared with the survey was done all the way back in 1959. Basically, they found out in comparison between the two, in the time frame of 50 years, 80% of the native habitat of Malleus silvestrii was lost due to agricultural development, animal grazing, a lot of overharvesting of the wild fruit, also a lot of top work of the native species into modern variety, and also a new apple insect problem in that region. Here's a picture showing this insect able to attack the Malleus silvestrii in the wild population. And also, this is a picture from the apple exploration all the way to the left, to the right, to the left. When they did an exploration in Kazakhstan, they actually see a lot of sheep grazing in the wild apple forest over there. The picture in the middle is not apple, but the argan trees from Morocco, but you can see the sheep really like the argan tree over there. Just trying to make a point here, okay? Here's a map showing the plant exploration of Malleus silvestrii in Central Asia by the Plant Genetic Research Unit in the late 80s into the 90s. Basically, there are 12 major sites. Most of them actually in Kazakhstan, but there's also site in Kazakhstan, Uzbekistan, also Tajikistan. Here is actually China. This is actually Xinjiang area. The survey was about native habitat was actually right here in this region. Here's a summary about tremendous amount of work on the exploration for Malleus silvestrii. Basically, there were 4 trips together, 12 major sites, and based on the phenotype they observe in the field, they actually select 44 they call Malleus silvestrii elite. They got the budwood and they bring back to the United States. They also collect over 894 seed accession of Malleus silvestrii from the wild. That including basically over 120,000 seeds together. And over 30,000 of those seeds was actually distributed to all the researchers worldwide. And about over 39,000 of the seeds actually still being stored at the Fort Cologne. And over 50,000 of those seeds actually stored at Geneva. In the last couple of years, we also have collected over 50,000 open pollinated seeds from Malleus silvestrii seeding block at Geneva. We call this the Botany of Desire seed for routine distribution. A lot of you probably read the book by Marco Pollan, Botany of Desire, and we got actually very routine requests from the reader because in the book mentioned that Phil Forstline is willing to give out the seed. So we constantly actually get inquiry about Malleus silvestrii seeds. And that's the seed. We don't give out seed from here, the original seed from the wild collection. We actually give out the OPC, so for the routine distribution to the stakeholder. In the last few years, we actually see an increase, a lot of inquiry actually from European country, England, Germany, France. Most recent was actually from Finland. Here to show you some of the picture from the exploration back in the 1990s. They basically, throughout the several explorations, sometimes they went back to the same location at different year to collect the material. Professor Dengelo is the major cooperator for all the exploration. He actually came to Geneva in 1992, and then the team went back in 93, 95, and 96. This is Elizabeth Dickinson. At that time, she already graduated, but she came back to join the exploration. Sometimes they also find very nice phenotype in the wild, and they will collect the budwood. They call that Malleus silvestrii elite, bring back to the United States. A lot of places they went to actually is really remote. Therefore, the only way they can get to is actually through helicopter ride. Here's some of the picture from the 1997 exploration in Sichuan, China. Before the exploration, Professor Li, who was from the Southwest Agricultural University, he was the leading authority on wild malleus material in China. He came to visit in Geneva in 1996. Then the team from Geneva went there in 1997, Professor Alwinko over there. Every exploration is a little bit different. One thing stand out in that particular exploration was there was an agreement that Americans were not allowed to collect the fruit in the wild directly. Only the Chinese scientists allowed to collect the fruit from the trees. Then the American able to participate in processing the material. The location they collect the fruit was actually next border to Tibet. So it's a very high altitude. So you can see Professor Alwinko trying to walk up the hill to look at the yaks. That's at the elevation about 4,000 meters. That's over 12,000 feet. Here's some of the picture from the 1999 exploration in Turkey. Basically, you have to do all kind of things when you're on the exploration trip. Some of them are fun. Here, basically, the route they took in Turkey, they start with the city in Izmir near Aegean Sea. They went up north, cover several locations, and then they double back once again to the north, coming back to Azir. I mentioned sometime we collect the bubble from the wild and to bring that material back to the US. Basically, for importation of Malleus germplasm, the bubble material, all the material need to go through, from foreign country, need to go through the USDA-ARS quarantine process before the material can come to Geneva. Every year, we receive between 10 to 20 accessions from ARS every year. In recent years, some of the material we got from ARS, that including Malleus cerevisiae, elite from the exploration. Also, some of the cider apple from northern Spain that was introduced by Professor Ian Merwin. Also, a lot of different variety from different countries. You can see basically from all over the world. Sometimes most of the accession, when they go through the ARS quarantine process, they take about a few years. But there are some extreme cases, especially this case. One of the Malleus cerevisiae elite from Kazakhstan was collected in 1996 exploration trip. It only released by ARS to us last year after 16 years in the quarantine. That just provisionally released. So we have to wait for additional 2 to 3 years before its permanent release to the public. So when it's all said and done, probably will take between 18 to 19 years before this material can be released to the public.
Why was that one so long in quarantine?
Sometimes baby just have some disease issues, so they need to clean up again, go through thermotherapy and the re-indexing. Sometimes just takes some time, some material very difficult. Now I want to switch gear talking about the preservation of the clonal collection at Geneva. Basically, all the variety and the species we have, we preserve them as a live tree in the field. Most of them, for the Malleus and Prunus, each of the session we have one mature trees. We start with 2 trees, but in general, after several years, we only keep 1 tree. For the Vitus, each session we has 2 mature vines in the field. For the wild Malleus and the Vitus germplasm, all the wild seed collection from the wild, we store them at -20 cold room. We have 1 cold room in Geneva, also 1 in Fort Cologne. A lot of dormant budwood for the Malleus, also the Prunus. They are under long-term backup storage using cryopreservation. Basically, store them in liquid nitrogen at Fort Cologne, Colorado. We also have a block of material in our collection. We call this the Malleus core collection. That including 258 accession. That's including majority of the common Malleus current day apple variety, but also other material from 42 other species and species hybrid. I want to do a Rubio here. Get some water. For the collection we have in Geneva, we have over 8,000 accessions total. For the Malleus collection, we have over 6,600 accessions, that including over 5,000 field accessions, also over 1,500 seed accessions. In the field, we have several different blocks. We have the B9 block, that's the Malleus core collection. We also have 2 other larger blocks, the M7 and the E7 block. That's all the material we consider a part of our permanent collection. We'll also have a K1 block. That's the Kazakhstan Malleus cerevisiae seeding block. They are on their own roots. It's different from the material in the 3 other blocks. They are actually on dwarfing rootstock. But for all the wild material, they're actually on their own root system. We'll also have another block. We call that W3 block. Actually, several different wild Malleus material. They come from Armenia, China, Georgia, Kazakhstan, Russia, and Turkey. We also have 1 block of material that including 5 different F1 population from a cross between Gala apple variety, crossed with 5 different Malleus cerevisiae. All these 5 Malleus cerevisiae, they actually have been shown to have a different disease resistance. So we consider this more like a pre-breeding population, or can use for mapping population for different purpose. For all our permanent collection in the field, every single tree in the field and every single vine in the field, we have a tag associated with each trees. Basically, will tell you which row and trees and what's the PI number, what's the species number, also the variety code. For the seeding block material, we usually have a tag for every 10 trees or so. Here to show you some of the picture from the Malleus cerevisiae wild collection, wild species collection. This is a picture from 2004, both the W3 block and also the K1 block over here in 2004. Here is a similar view in 2007, 3 years later. This is a Malleus cerevisiae, different species. Here is all the Malleus cerevisiae from Kazakhstan. Well, a little bit wild vitis, different wild species on the corner there. Here's the view from the Kazakhstan K1 block in 2012 last year. Because they are on their own roots, they are not on dwarfing rootstock. The material basically are now over 20 feet high. Here's a picture of Phil Forstline, the first curator, together with Professor Herb Alwenko, looking at Malleus cerevisiae material in the field. They spent a lot of time on those material. Here's some of the pictures showing some of the interesting stuff we have in our collection. For example, we have some of the large fruited Malleus orientalis from China. It could be hybrid. We're not 100% sure. This material, they are very large fruit, so they have some ornamental potential. We also have some material called Malleus transitoria. They're also from China. They are very dwarf material, also very spreading, very different from most of the wild species. This is one of the Malleus hybrid material called butterball. It's a beautiful ornamental material. It tastes terrible though. We also have a small Malleus nursery we use for our propagation purpose in our collection. Basically, this shows you a very standard Malleus material in our permanent collection. We planned over 150 new accession last year. We also have a small stew bag for rootstock propagation. Here's a very interesting material. It's a very thorny juvenile material from the wild Malleus hoopiensis from China. You can see all the thorny material. Usually you will never see that in commercial apple. These things can puncture through the tire, basically. For the vitis collection we have, basically, most of the material we have, they are wild species, also a lot of species hybrid. That means between Vitis vinifera crossed with different wild species. Therefore, they have a cold tolerance, able to stand the weather in Geneva. Most of the Vitis vinifera material, that's the common wine grape, table grape, and also some raisin grape. Those are all housed at Davis Repository because the weather is better over there. We have over 1,300 accession in the field and about over 80 C accession in the cold storage. Here's just another view of our basically main Vitis collection in the field with all the Malleus collection in the background. Here's also another material we have in our collection. It's a Vitis hybrid. It's a seedless material. All the best tasting material we have in the field. Usually by end of the season, it just disappeared. Here's another interesting material. It's a wild species actually from China, Vitis zakai. This material is actually very unique leaf morphology. It's a very deep wedge material. That's only in 2 species among over 60 species actually have this type of morphology. For our Tertiary collections, actually relatively small collection. Basically, Tertiary is a diploid species. Actually, it's a hybrid material from sweet cherry crossed with some of the wild tetraploid material. For example, the Fucus, the Punus fruticosa. We have 130 accessions in total. Tart cherry is actually a very small industry, only $40 million in the US. Compared with the grape industry is over $3 million. Apple industry is over, $2 billion, $3 billion and $2 billion in the US. Most of tart cherry actually originated from Central and Eastern Europe and also from Russia. Majority of the tart cherry accession we have actually came from the Michigan State tart cherry breeding program that's in charge by Dr. Amy Arizoni. We've done a lot of characterization for the colonial collection we have. Basically, all the accession PI number, all the species number, species name, the varietal name, the general description of the material, the source history, the pedigree, if there's available, also a lot of digital imaging, actually all stored at the GRIN database. We also carry out a lot of phenotypic, also genetic characterization, also some other work such as the chemical composition, the ploidy, measurement, disease resistance, growth habit, morphological trait, and the phenology trait, some of the description about the production, also microsatellite marker, some of the information we have, all stored at the GRIN database. In the future, hopefully, we'll have more GBS information, the genotype by sequencing, marker data, also more food quality data, also the sequencing data in the future. All the information basically is searchable at the GRIN database website. I want to talk about a lot more about the wild Malleus material, a lot of evaluation we have done in the last 20 years. Tremendous amount of disease evaluation work has been done in the last 20 years. Evaluate against different fire blight, scab, the blue mold, post-harvest decay. Also a lot of work on mapping of resistant genes from the wild sources. Also a lot of work on drought resistant evaluation, also cold hardiness. Also a lot of genetic diversity study, population study. Also using the genetic marker, trying to determine the core collection. Also using genetic marker, trying to identify interspecies hybrid, also duplicate in our collection. We also have a lot of, I mentioned before, some of the mapping population that could be used for mapping different kind of trait. Here I want to give you one specific example about disease resistant evaluation against blue mold. This is work between the Appalachian Food Research Station of USDA at the Currinsville, West Virginia, together with the researcher in Volcanic Center and the PGRU. This particular accession actually is being shown to be resistant against the blue mold, and they came from Kazakhstan. In 2011 and 2012, we collect fruit from 101 individuals from this F1 mapping population. Then we inoculate the fruit with penicillin expansum, and then we measure the lesion size 7 days, sometimes 5 to 7 days after the inoculation. There's a huge environmental difference between 2001 and 2012, but the disease rating across the year actually highly correlated. The QDL mapping for the blue mold resistant actually being mapped resistant to linkage group number 4. Here just showing a picture. We're collecting the fruit last year in the field for the inoculation experiment. Here to show you an example of 11 fruit from one susceptible and the one resistant progeny. You can see basically after 5 to 7 days after inoculation, there's significant difference in the lesion size between the susceptible material versus the resistant material. We also use different genetic marker. In this particular example, we use 9 microsatellite marker to study over 1,900 Malleus accession we have in our collection. We're able to identify 238 Malleus domesticata and also 10 other Malleus accession. They actually share the same genotype based on 9 SSL marker with at least one other Malleus domesticata accession in our collection. Once we find anything with a similar genetic profile, we're also trying to look into the fruit image, trying to confirm the genetic study result. In this case, we're able to identify a total of 23 spore family, that including 104 accession in the collection. Also, there are some mistakes. We identify also some duplicate we're able to identify from this study. We also carry out a lot of horticulture and food quality trait evaluation. In the early year, our unit actually devoted a lot of effort into cryopreservation method development for the Malleus and the Prunus. And also, together with Fort Cologne, they did a lot of study on long-term survival rate of the cryopreservation bud material. Also, in the last couple of years, we focused a lot on the digital image of the fruit, the leaf, and flower. We upload a lot of image into the GRIN database. We're also involved with polyphenolic and also antioxidant capacity study of the Vitus. Also, we're working on the Malleus and the Tertiary right now. Also, juice quality and some specific example like the red flesh apple, and also fruit abscission rate of some of the Malleus accession we have. Here, just show you a different digital image we have for different material in our collection. Sometimes we still have some shadow. We're trying to improve the quality of the photo in the near future. Here's just another example of a genetic study of a red flesh apple in our collection. This is study including over 100 accession. Basically, they have different kind of red flesh. You can see for the different pattern of red flesh, they have some of the red flesh actually localized in the calyx area. Some of them actually associated with the vascular bundle. Some of them just throughout entire cortex, but not including the core. Some of them just focus on the core region. And some of the material actually throughout entire fruit. So there's a lot of different color, different pattern, also different intensity among different accession. We also look at trying to use PCR, trying to identify the MIP10, which is transcription promoter, that previously has been mapped to linkage group number 9. That's strongly associated with the red flesh fruit among all the genotype. But this particular promoter is not neither sufficient nor required for all the red flesh in all the genotype expression. Also, nearly all the red flesh material we're able to test it, actually all could be traced back. Probably a single variety, Izuwaski Anna, actually is a wild, potentially probably a wild material of a Malleus cerevisiae from Central Asia. So all the red flesh available right now is actually all could probably trace back to this single variety. Last year, we also upload a lot of ploidy information based on flow cytometry, also some of the information based on historic information available from the GRIN. We upload 682 Malleus domesticata and Malleus hybrid ploidy information into the GRIN. That including 554 diploid material, 17 chimeric, 91 triploid, and 20 tetraploid. We also have flow cytometry information for over 664 wild Malleus accession from 53 different species. The ploidy is a little bit more different. Some of them, they have diploid, triploid, tetraploid, all the way into pentaploid within the same species. Also, we have microsatellite marker data for basically all the permanent accession we have in our collection. So in principle, we should be able to infer the ploidy level from the SSR marker in the future. Since last year, we also initiate the Malleus genotype by sequencing effort for our Malleus collection. In this study, we're including over 1,950 material from our collection in the study. That's including over 1,400 Malleus domesticata, a lot of different wild Malleus species, all including in the study. We use 2 enzymes for restriction. We have 21 play in total, and we're just in the beginning stage of the data analysis. For each play we're able to obtain between 100 to 100 medians good rate per play. So basically, we're doing a lot of sequencing information from this study. In some of the initial analysis we're using Golden Delicious Genome as a reference genome for comparison in the analysis. In that particular study comparison, about 40% of the sequence actually could not be mapped back to the Golden Delicious genome. Actually, 40% of the sequence we identify from the GBS study actually never been known before. Also, in the future, we're trying to make comparison without any reference genome, so we're not going to create any bias against, especially the wild Malleus material. Hopefully, in the future, we can use the data trying to study a lot of different questions. Domestication issues, still there's some debate over there. The breeding history, the gene flow, the evolution, also gene trait association. I hope something we can do in the future, and also other issues related to the Malleus genome. Next up, to switch gear a little bit, talking about distribution for the Cologne collection. Every year we update our catalog for apple, grape, and tart cherry that's available online. Basically, all the distribution are free of charge to the stakeholder. Here's a summary table of some of the number of samples we distribute in the last 10 years. As you can see, back in 2007 and 2008, in combined, we distribute over 11,000 samples in those 2 years. In the last 2 years, we distribute over 9,000 material per year in the last 2 seasons. So that's a lot of work, a lot of material. Here's just a picture showing we're collecting dormant bulbwood from the Malleus cerevisiae seedling from Kazakhstan for a drought-resistant study we're cooperating with. This is about 3 weeks ago. Believe me, it was very cold in the field. As I mentioned earlier before, another purpose for the distribution I see is actually trying to promote the value of the germplasm material we have in our collection. Therefore, we carry out a lot of outreach, education events, also a lot of tools coming to a Cologne collection. Basically during the springtime, the summertime, also in the fall season. So far, not too many people come in the winter time. Every year, for example, the plant breeding courses, they will come to our collection in the fall. They will come to Geneva. I think when they come to our collection, they probably have the best time. They're all very happy. They carry our fruit back to, so they're all very happy to come to the collection. Last year, also the summer scholar student at the station also came to look at the collection. We're able to have Dr. Helber-Albenko actually talk with students about all his exploration experience. We also carry out a lot of germplasm exchanges and also visit with other gene banks throughout the world. For example, this Miss Yoon Young He. She is the grape curator and also molecular geneticist for the South Korean Gene Bank. She spent about 3 months with us last year. She's back in South Korea. In the future, hopefully, we're going to start exchanging material, especially the wild Vitus material between the two gene banks. Here's kind of in the summary, basically the short-term goal we have for the Cologne collection in the next couple of years. First of all, we'd like to continue digital imaging of the collection. We can always take in more photo. Also, we are trying to finish the evaluation of Vitus phenology. We already have 2-year data. We're going to do that for additional 2 years. Also, we're going to start the Malleus phenology evaluation, hopefully for the next 3 years. We're continuing on the food quality, also in nutritional evaluation for the Malleus and tart cherry, the work we started since last year. We're also going to carry out a gap analysis for the Malleus, also Vitus germplasm material, using different geo-referencing data, the climate data, also genomic data when it's available. Once we're able to finish the gap analysis in our collection, we intend trying to begin collection for more systematically for the North American Vitus material which we think we really should focus on for the next couple of years. Also, potentially, we're probably trying to collect additional material from the North American Malleus species. We have 4 different species in North America. We're going to continue trying to apply the GBS marker for the Malleus and also tertiary genome analysis, probably more trait association analysis, and also probably potentially allele mining for a lot of wild Malleus and also the Malleus and tertiary material we have. Also, we're cooperating with a lot of different people. We'll continue on evaluation of a lot of wild material of the Malleus, also the Vitis material. Based on those evaluation results, we're going to try to select some of the material to be included in our permanent collection in the next 5 years. Some of the long-term goals we have for the colonial collection, first of all, we hope we can carry out genetic study among our gene bank together with gene bank in other countries. Hope that will lead to future exchange of material in the future. Also trying to exchange and exploration maybe the wild Malleus and also Vitis material, especially in China, potentially in Turkey and other places. We would like to complete the long-term backup storage for all the Malleus and tertiary accession, and trying to develop a very good, reliable long-term backup storage method for Vitis, which we don't have right now. Also, additional genotyping and phenotyping of all the wild germplasm material we have in our collection. We'd like to reduce the disease level in our field collection. Hopefully trying to look into long-term, trying to develop a new strategy for long-term preservation of the colonial material we have, because we're probably facing potential limiting space issue, also resources issue in the future. Finally, I want to acknowledge all the people involved with the program for the last 30 years, especially a group of guys I work with every day. We call them the A-team. There are 4 guys that actually work on the collection day in and day out, basically doing all the work. And also other people from the PGRU, and also former curator Phil Forstein, Professor Alvin Ko, Plant Pathology Department, Larry Robinson, Geneva Facio of our unit, and also folks at Kearneysville, Gail Volk, Chris Richard from Fort Collins, other people at Fort Collins involved with the long-term cryo backup storage, and also all the folks at PGRU and GGRU, and also the field research unit of Cornell University at the Geneva station. Throughout the year, they gave a lot of help. Also all the folks at Cornell University. And finally, the funding support from the National Cereal Project and also the AE9 Regional Experimental Station funding. Thank you. The question was, has the Apple genome been sequenced? How we, in that case, how I refer to Golden Delicious, how we're going to use that? Yes, there's a published sequence based on Golden Delicious. That's what we use for our comparison, some of the material, the GBS data we have. But I'm not an expert in the area, but the discussion is, because throughout the domestication history, if you simply use Golden Delicious genome sequence as your reference genome, we do have a lot of wild Malleus material that's probably not involved with domestication of Golden Delicious, the Malleus domesticata. So if you use Golden Delicious as the base for comparison, you're going to be biased against a lot of wild material. So we're going to try to use Golden Delicious sequence as a comparison. But also we're trying to do other comparison without any Golden Delicious genome sequence, so we're not creating any bias. We're still working on that. A lot of other issues involved. For example, triploid is a very serious issue. Polyploid is very difficult for GBS analysis right now. Most of the methodology, my understanding is developed for diploid. So we do have a lot of polyploid material, and also especially the triploid material that can create a lot of problems. So we're still working on that. We have a problem with Phylloxera disease. Yes, we do in the field. We do have different disease. That's always a concern. But it's very difficult because it's open field collection. so it's never been clean. And we don't really have resources to testing material all the time, so it's always an issue. I do mention that. For the foreign distribution, all the material we send out have to go through the quarantine process of the receiving country. But in the United States, at least for the Malleus, that's not the case. The question is, the question is if any plan for mutagenesis analysis of some of the collection. There's some research involved with for the Vitus material by the GGRU folks. They are doing some mutation looking for certain mutant. They are looking for that, some of the material, but not for Malleus, no. Yes, you're right. The main disease problem we have is a fire blight. Potentially, of course, right now we rely on chemical control, but that's always difficult, chemical control and also plus the pruning. Hopefully, potentially, we can look into some other strategy, maybe cover crop, for example. That's a possibility. Trying to hopefully can reduce the disease level in the orchard. But it's always a balance between how much resources we have, how much we can do. We're pretty much stretched very thin right now for the work we have to do. So always appreciate more help.
This has been a production of Cornell University, on the web at cornell.edu.